the data straight from genomics. How do we go from that to something we can work with, something we can start to visualize in IGV, and then actually do some downstream analysis, count our signal in genes, compare our signal in those genes across conditions. So before we go any further in the course, I just wanted to bring attention to our GitHub. I'm sure we've seen this already. So if you have any problems and it's not in class, you want to still ask them, maybe get some responses. We have this help page here. Again, if you just go to getting help, I think you need to sign up to GitHub to actually ask for it, but then you can ask questions here, either things which aren't quite right in the slides, or, you know, just confusion about some of the things we've said. You can ask it here, and we try and leave these open so that the next person who comes and asks the question, why doesn't my IGV accept a BAI file? You can go and look and see what the answer they had. So, you know, we're getting complex now. If you have questions, you don't come across these questions, you have them after you've been through the material, just click here, ask us a question. And, you know, Matt's really good at going through and trying to respond to these. So today we've done all the basics of R, and now we've introduced ourselves to Bioconductor. We looked at how we handle sequences. Both our FASTA and our FastQ are composed of sequences. We have our genomic intervals. How do we represent regions of interest in the genome? How do we then assign score to these regions of interest? How do we export that, import that? How do we export that, import that, manipulate it, log it, you know, do arithmetic on these genomic scores? And then we looked in our last session yesterday at genomic features. How do we represent gene models? Okay, and gene models are simply where the exons are, where the coding regions are, the whole bounds of the transcript, how transcripts sum up to a gene, right? All this information is stored in gene models, which we can either get in a GTF or GFF, or we can get internally from Bioconductor TXDB objects. But whatever happened, we knew we could bring them into Bioconductor and start to operate them there. We also looked at, and it'll be very useful, is the org-db packages. And this is just how we go from a entrees ID, one of these numbers we see, or an ensemble ID, to something we can really read, a symbol, a gene name written out in full. Okay, so now we're in this session. I'm going to go back to the beginning, and we're going to start to look at the first type of data you'll actually get out of genomics, which will be the FastQ, faster with qualities. So as Connie said, and I'll just repeat a little bit about what she said here, just to remind us, we can now massively parallel DNA fragments, and these are fragments, not long stretches of full mRNA. That is the reserve of places like PacBio, and we're going to hear about that later on in the course. But for DNA fragments, typically things like RNA-seq, ChIP-seq, whole genome sequencing, when you're fragmenting your DNA, we can use Illumina, which was originally called Solexa sequencing. This allows us to profile things like the abundance of particular DNA sequences, and that may be either cDNA sequences. We take our full RNA pool from our sample, we reverse this into cDNA, first transcribe this into cDNA, and maybe we'd fragment that and we would sequence that, and that will give us a representation of what cDNA, hence what RNA was in our sample. With this full genome sequence, so with this FASTA and our reference sequence, we should be able to align these little fragments, these tiny bit of sequences we have to the full genome, and then putting it in context. And with things like our transcript models, we can then get an idea of, all this sequence went to this gene. We have a lot more sequence in this sample than that sample. And then we have our differential gene expression, and we will go and do our biology on that. I think Connie described this so much better than I will, so I'm really not going to bother. But I will summarize it again. With our fragmented DNA, be that cDNA or DNA, from an IP or from your whole genome sequencing experiment, this gets attached to the Illumina flow cell and through a process of actually, what's the word for this? Let's have a look. I guess amplification, but we actually attach, after a process of amplification, we then get into the sequencing, and for one base at a time along our sequence, right? So a copy of this DNA we have here, we put in, we will attach a colored base, one base at a time, and this is called cycles. So for the first cycle, we look at the first base, and these bases we attach are fluorescent. And every time we add a cycle, or we add this bases, we can tell, okay, an A got added to this sequence. So we know it's an A at that position because it fluoresced red. A T, right? So this bit of sequence growing here, we'll have a T perhaps. And over time, we keep imaging every cycle, and we get an idea for every sequence attached to our flow cell, what the sequence was in these fragments. Okay, so really what we end up with is a series of images of cycles. In this first cycle, we had a green, that's a T. In the next cycle, we had an A here, and then another A, and then another G. So we can use this fluorescence and imaging to give back to us some reasonably confident sequence from the ends of these DNA fragments, right? So we'll just usually sequence just a portion of these. So all those images, all that adding bases, all gets turned into this FastQ. And this just represents, as we saw before, the sequence from the end of a fragment. And we have one record here. So this is one fragment sequence, just the end of it. We can tell where it was on the flow cell by looking at this information here. This tells us exactly where it was. We know that this was the machine. We know that this was the read number. Here's the sequence. And here was the difference between FASTA and FastQ, is here we have the qualities. Okay, because you can imagine as this process of adding your DNA bases, imaging them, adding more bases, imaging, as you move through these cycles, the chemistry is a lot better than it used to be, but the chemistry slowly tires, and you're slowly going to get worse quality as you move along the read. Okay, so here we have the base pair positions across our read. So we'll often refer to these as cycles. This will be cycle 1, cycle 2, cycle 3, cycle 4, cycle 5. So Illumina sequencing, FastQ format, which comes from this, is absolute standard for sequencing. I can't even remember what the older formats would have been, maybe faster within a quality file separated. But all this can be handled in one of the oldest packages in Bioconductor, short read. And we're going to be able to use lots of the functions we saw earlier on for working with FASTA by way of the biostrings package. So it's really short read is going to be able to read it in, allow us to deal with the object, but we can use all this functionality we saw in biostrings to start to operate on these reads. First things first, we need to install. If we want to install any Bioconductor package, we just need to go Google short read. It will pull up the page here, and we can just copy and paste this bit. It checks that you have BioC Manager installed. If you don't, it installs it, and it will go get the rest of the package. Okay. Once we have short read installed, we can then make use of all these functions. So in this particular session, we're actually going to grab some new data from the ENCODE consortium. So we're going to grab some raw sequence data for FastQ in FastQ format, generated from here in an RNA-seq experiment. So it's going to be the cDNA copy of the RNA fragmented, and then we've sequenced that. So you can actually find this, there's a link here apparently. You can come to here, and as always with ENCODE pages, they're very nice in that they include the raw data, FastQ here, or in fact the process data. So we like to use ENCODE in our courses because we can pick the data of various parts of processing and see how we're doing using the methods in this course. If you want, you can also click there. I think it will download the actual direct link for you. Download the file directly. But in order to get us going at the beginning, we're going to work with a sampled part of this file. And I've actually just taken the first 100,000 reads in the file, and I've created a file which is in our data directory, sampled, whatever. This should allow us to quickly perform some processing and analysis while you're kind of working along with us. We will show you as we get towards the end of the session how you can use short read for full data sets. So we do use internally short read to do some of our processing. You just need to set it up to be a bit more memory friendly. To get us going, I'm going to start with this sampled file, but at the end, I'm going to show you how you could go from full-size, multi-gigabyte size files. I'm also going to allude to another program we actually wrote, which will do this very fast. But this is a really core bioconductor, so I'm going to show this first. Okay, so the short read package allows us to import FastQ, much like our track layer package has allowed us to import things which were aligned. The short read package is going to allow us to deal with FastQ. We just need to use the cleverly named read FastQ function. And this is going to return a short read Q object, and that contains the FastQ sequence, the qualities, and all these identifiers. So we're going to import all the components of this, and as you know, there are only 3 components we really care about: the ID of the file, the ID of that particular sequence, that read, the actual sequence itself, and the quality associated to the sequence. So we read that in, and we get our class short read Q, short read quality, and a short read from the short read package. And if I type FastQ into the screen, it's not really going to show us much. It doesn't want to try and show us the information in the file yet. It just tells us the class. It tells us how many reads we have. Okay, so we know we had 100,000 because I sampled it to that. And it tells us the width. Okay, so here, width, we can think as bases. Remember, I was saying cycles are the same thing. Every time we add a base, that's a cycle. So we call it 76 cycles here. We can use our very familiar methods. Everything in R, not everything in R, but a lot of things in Bioconductor will allow you to apply the basic methods to them. So if we apply length, we'll get back, as you might expect, the total number of reads, the length of the object, just as we can do for vectors, GRanges objects. We can also use width. So we actually used this for DNA string set objects before, and we used it for GRanges objects. So here, if we give width, rather than say number of columns or something, if we use width, it's going to give us the width, the number of cycles for every read. Okay, so here, they're all going to be 76 because there's been no trimming, there's been no editing of the read. So they're all the same length. In fact, we could have told that here. It told us that we only have 76 cycles. If I had reads in my data which were 40 or less, it would show the multiple cycles, all the unique numbers of cycles I have. We can subset these short read Q objects just as we've done for vectors too. So here we can either use 1 to 10 or whatever we would, however we would like to specify our index. So we can do FastQ. Yep. FastQ square brackets 1 to 10. I just have the 10 reads, and I still have my full length of the read. As we have seen with things like genomic ranges, because this isn't a classic table or really a classic vector, it's more like a list, right? Every element will have, every read will be an element, and we'll have an ID, a sequence, and qualities. So we need special accessors to be able to access it, right? So thankfully for us, the short read package provides some accessors, and they really just get at those 3 components we really wanted. If we want to retrieve the sequence of the read, we can just use sread in our brackets, give it the actual sequence, give it the object, this short FastQ object, and we'll get back the reads. If we do quality, we can do retrieve the quality of reads as ASCII scores. Remember, these aren't numbers, they're meant to be probabilities. So it's the probability of this being correct. Higher the better, but to save space, we've encoded these as ASCII scores. If we use this qualities quality function, it's going to give us back a string of ASCII scores. Finally, we actually have the ID function. It's not IDs, it's ID. And that's going to retrieve the IDs of the read. So all these names we have, and that may be very useful for us to be able to match up across files. Maybe I want to know where that read ended up in my BAM file, or maybe I want to use that information. Sorry, I'll skip back. Use that information to know the machine or even the position in my flow cell. It might be I have a problem spatially in my flow cell, and I could identify that by looking at the read ID, working out where these reads were, and seeing if I have a problem in a particular area. First thing we'll do is retrieve all our read sequences. So we just use that using this sread function on our shortReadQ object. If I do sread FastQ, I get my sequence of all my reads. And then I can look at the class, and hopefully it's already something we recognize. It's a DNA string set, okay, from our biostrings package. So we already saw DNA string sets with FASTA files. Okay, so we saw that we store the FASTA file in a DNA string set. Every element there was a chromosome. We also saw DNA string sets when we extracted sequences from underneath peaks. Right, it gave us a sequence, a DNA string for every peak, and it was the sequence underneath it. Here, once we've extracted the reads within our shortReadQ object, it is again a DNA string set. So that's great. We know how to work with DNA string sets. And it means then we can use all this functionality we saw in the biostrings libraries now on this DNA string set object. Okay. So if I want to do something like the alphabet frequency, which we saw for the FASTA file, we can run that on a DNA string set. I just need to do alphabet frequency, give it my DNA string set object, and I have this here, the number of times an A has occurred in this read, a number of times a C, a number of times a G, a T, here an N. So clearly these first reads are mostly N, but already I have this really efficient way taking all my reads and calculating, what is the distribution of different bases across the whole of my read. We can also extract the IDs for every read using a very similar trick. We just use the ID function. And it's going to return to us here the IDs of the read. But it's not quite it's still a biostrings object, but it's not going to be a DNA string set. It's not an amino acid string set, an AA string set. It's something even more basic, is just a biostring set. Okay. And this has similar information with, and then the actual sequence here, but it's not quite the same as a DNA string set. But we can still operate it, subset it, and retrieve information from it. One thing we could do, like we can do with DNA string set objects, is actually just use our as.character function on our BS string object and just turn this back into a normal character where we can do all the operations we could on character objects. Okay. So a key fundamental difference between FASTA and FastQ are the quality scores associated, contained within the FastQ. So as we saw, these are encoded as ASCII characters. So we have some map somewhere which is taking our minus log 10 probability of a base being wrong. So larger scores associating more confident base calls. it's taken that and it's encoding that in our FastQ to save space. Right? Like I said, if you wanted to go really understand exactly the calculation and the quality, you can see a little map here, and they tell you for the different standards of quality. It was complex before, it's not so complex now. You can actually look and you can see that, you know, this value here maps to this score, this value here maps to this score. And we can see here clearly hash right at the bottom. And we saw a lot of hashes just now when we were looking at sequence, and this tends to be a very poor quality score. Did I leave the slide? No. So just to show you that again, I think when we looked at our first reads, they were all hashes at the end. Yeah. Like I was saying, as reads over time, you tend to get less high quality at the end of your read. You run, the chemistry starts to tire. Okay. So we can extract these qualities for every read using the quality function. Okay, and it's going to do just like the S read and just like the ID. It's going to return us a new object this time, though. It's actually a short read, a FastQ quality object. It's from the, is now associated to the short read package. This is very much like a biostring set object, but because these qualities are so particular to FastQ, you know, it's been adapted here within the short read package. Okay. If I just type in quals and we then look at this in the screen, we can immediately see lots of hashes, so lots of low quality reads at the beginning. We have our length, we have our total. Okay, but actually looking at that, apart from the fact that I just went and looked up what hash was, actually that's not too useful, right? I could say it looks bad at the beginning. I don't know what these are. I don't know what this means. So we need to be able to translate that for us. Okay, so we can find out how to translate these ASCII characters into their corresponding -log10 p-value bins using the encoding function on our FastQ quality object. So we don't need to go and map back one at a time from the Wikipedia page. We can actually use this encoding function, pass it in our qualities, and it reviews this and it tells us the quality scores associated to each ASCII code. Okay, so like I just showed you, hash was right at the bottom. Actually, this matches obviously what we just saw on the Wikipedia page, right? But now we have it in our, importantly, we have it as a named vector, right? So this means that actually we could use this to maybe map those quality scores to, or those quality ASCII to the actual score associated to them. Okay, so first we just need to dig into our FastQ quality object a little bit to get a score. Okay, so we can subset the FastQ quality index quality object either by using, much like a list, either by using a single square bracket, which will still bring us back a FastQ quality object. Just like if you use square brackets to subset a list, you still get a list back, but it's just the element you've selected, right? Or elements you've selected. If we actually want to get to the underlying BS string object, we're going to have to do the double square indexing, double square bracket indexing. And that's actually going to get us underneath the FastQ quality object and actually at that BS string object itself. Okay, and you can see that here when I do quals, single square bracket, I go from being a 1,000 long to just being the index I selected, but I'm still a FastQ quality object. If I do double square brackets, I can actually get at that BS string object underneath. Okay, so now we can try and split up this read, this qualities in this read into a vector, and I can use that vector maybe for the translation. So I think we haven't used this function yet, but it's really cool function, so we can use it here. StringSplit, it just allows us to take a character. So here I'm turning our BS string object into a character, and I can split that character by any character vector I want. Here, I'm going to split it by nothing. Okay, so it's just going to break up our character vector, which was this long stretch of hashes, into separate values So now I have a vector rather than one value of all the with all the hashes in. I've split it up into a vector of around length, I guess, 76, and each value is a hash. Okay, so that's what I have at the end here. Now I can just use that vector, this vector of hashes. I can pass it to this map, right? So here I have the names, and here I have the values. This is just a named numeric vector, right? So if I pass in my vector of hashes, it's going to look up the hash, and it's going to find out it was 2, and it's just going to give me a string back of 2s. Okay. So I can, if I wanted to, write a little bit of how I may turn my quality into a score I can actually translate in my head or understand in my head. In reality, we're not going to do this. It would take a long time to cycle through this. It's nice to do, but there are functions available in the Shortre package to do this for us. And one function which is very useful is alphabet score. So alphabet score goes and does exactly this translation for us, because you can see we're going to get the same value. But if I apply alphabet score to my qualities FastQ quals object, it's going to give me back a vector of scores. And if I look at the first one, 152, if I did that translation we just did over these slides, we'll get the same number. Right? Fact is, this will be orders of magnitude, hundreds of times faster than operating on a file. What's really nice is that with this one function, we can get how reliable a read is overall. And then as you're going to see in the exercises, you will plot the distribution of your scores, and you want to see, are any reads really sticking out for low quality? We're going to show this, I think, perhaps in the exercise, so it's kind of safer there. But another really useful thing to do is not just to look at the sum quality across a read. Okay, so maybe a read overall is bad, it didn't sequence well. But like I said, reads will get poorer quality over time, over cycles being the first cycle, the first read we sequence, sorry, the first position base we sequence. You know, as we move along, all these reads, quality will drop off over time. Okay, and we want to capture that. Oh, well, you would just divide or normalize by the width, right? So we can get all the widths at the same time as a vector, and you could just do a vector division at that point, right? So you could have sum of the scores divided by the read length. You would run, sorry, this is a question from Nathan, if anyone's wondering what I'm talking about. It's a good question though. Oh yeah. But I just want to talk it through for everyone as well, Nathan, because it's a good question. So actually what we're going to get, so Nathan's asked, okay, we can get our sum quality, but not necessarily all our reads are the same length, right? So if I do this alphabet score, I'm going to get my read score. That's good if all my reads were the same length, and we knew that in this case, right? But we might not know that, or we might not be sure. So the first thing you would do is see here, I have 76 cycles, but I can also extract, right at the beginning, I think we saw we can extract width. So if I extracted all the widths and all the scores, I could just do scores divided by read lengths, and I would normalize to read length. That's a good question, thank you. So the next thing we were worried about was, you know, quality over cycles, right? So maybe our quality is dropping off. And actually this is kind of important because we might want to just get rid of the end of the read. We might decide it just drops such bad quality, we can't trust it, and we'll just try and remove it. Okay. So actually to get our qualities, this FastQ qualities object, what is it called? FastQ quality object, into something we can really work with, we have to do this as we give it the quals and we tell it we want it to be a matrix. And then it will just give us a matrix of samples, reads, and then quality. So it's going to be a massive matrix. But from there, we can start to do plots quality over cycles. Okay. I just show the first one here, row sums of the first one. So I can show no matter which way we did this, manually ourselves, using alphabet score, or turning the qualities into a matrix, we're still going to get the same value. Okay, this is in the exercises, so have a look in there because it's a nice example. The Shortre package has many functions available to collect useful metrics. Alphabet by cycle, that's another really useful one. So it's going to quickly summarize for us across all the reads how many times we had an A, a C, a G, or a T. We just need to give it the sequence of the reads. Here, it's what we got returned from the sRead function, the actual DNA string set object of the reads. We can use that, and it's going to return. I'm only showing you the first four, but actually it returns A, C, G, T, and all this IUPAC code, R, Q, whatever, and then eventually N. And then you can see working across cycles, so this is the same as position in the read. We can get this idea of, are we getting a bias across time, across cycles for particular bases? And actually a lot of your samples will show some bias. It's always important to compare across all the samples you're looking at in order to, it's really important to okay. So then the last thing we can do is our new alphabet by cycle, but we can actually apply it to our quality scores too. And then this will just give us a reasonable idea that we have, for instance, on this cycle, we have this many of this score, this many of this score, this many of this score. This can also be very useful because although this is still our hash or our quote or our exclamation mark, we could actually translate this into a score and then use that for plotting later. Something which is potentially very useful for us is to actually look at whether or not we have something in our reads which is maybe occurring more than we might expect. Okay, so we can do this very easily. I think Matt would have shown this in the first few sessions. We have this table function, and this will just tell you the number of times I see a value in my vector, and it will just give us back a table. This is the number of times for this particular value. So we can do the same thing here with our sequence of reads. So we just need to give it our result from our sRead, our DNA string set object, give it the table function, and we can get our read occurrence here. And first off, we can see that we have lots of reads, which are just all, in this case, totally blank. So out of our 10,000 or 100,000 reads, we have 403, which are all blank apart from the first position. Okay. And if I wanted to here, I've just sorted them by their read occurrence. So we should be going decrease equals true. So this should be the most occurring. And you can just see that there. Potential problems in sequencing. One potential problem is that you go in with a library where you just don't have enough complexity. So for whatever reason, maybe you started with a very low cell number and you actually haven't captured too much RNA, or which has been then been turned to cDNA, or you've done a chip, an IP, and it wasn't very efficient. You didn't really pull down much. At that point, when you sequence it and you do things like PCR, you're going to get very biased in your amplification, and you're just going to see lots and lots of the same sequence occurring in your data. Okay, this can be an issue, and it's always worth, if you have multiple samples, just seeing your duplication rates of your sequences, and if you see one sticking out, or if they are more duplicated than perhaps your friends, your RNA-seq you're comparing to, your chip-seq you're comparing to, maybe we have a problem. So we can actually use this SR duplicated function, and it's a really optimized function here. It's going to hopefully be very fast. So if you use SR duplicated on our FastQ object, so we can just give it the whole short read Q object, it's going to tell us a true, false, whether a read had already occurred in the list. Okay, so it's going to tell us whether or not a read is considered a duplicate. Importantly, notice the first time the read appears in the file, it is not a duplicate, but obviously after that, the second, the third, the fourth is all going to be marked as duplicates. So with that combination of table right here, it's just going to tell us the most common sequence for a read. But now if we use our duplicates with this table, it's going to tell us how much of our total reads were duplicates. So that's the true, and how many were unique sequences. Okay. So, this really depends on your data you're looking at. So don't be worried. You will have duplicates in your data, especially if you're doing RNA-seq. You only have a limited amount of space you can really sequence. It's just the RNA. But it's always worth keeping an eye on this and capturing this information at this point when you start to look at your sequence data. So the short read queue short read package has this QA function which accepts a FastQ file and returns a FastQ QA object. Okay, so we just pass in the path to our data sampled and then the FastQ.guz. We have our my QA here, and this is now actually contains all the quality assurance. It contains lots of quality information on your FastQ data. If you want to, you can make a report from that. And it's going to go and it will write out, and we should have one, maybe we don't produce this, but you can run this yourself. It will produce a nice little report summarizing some of the information we've seen now as we've run through this example. And if you want to go look at any reports, if you ever want to just look at an HTML file from within R, because this function returns where the HTML file is, we can then just use the browse URL function in R, and it will go and open up that particular HTML, in this case, the quality report. So I'm not going to show you this. I'm going to show you our quality tool at the end very briefly. So I think it's a lot faster than this short read one, although the short read one's perfectly acceptable. When we observe some of these things, like I showed you, we worry about quality dropping off over time. We may wish to actually remove some of the low quality bases before we actually go and do some alignment in the genome. Okay. The aligner I'm going to show you in the next session doesn't require you to do this trimming. Right? It does this for you as part of the alignment process, but for completeness, I wanted to show you this here as well. So the trim tails function is going to trim reads from the 3', removing bases which fall below this desired quality. So working from the 3', it's going to do this. So it needs to accept our short read queue object. It's going to accept the minimum number of successive bases required to be below the quality cutoff for trimming. So we need 20 bases in a row to be below this before we start trimming. And then this is the actual quality score cutoff. Okay. So you're going to look, most often you will review your data first. And then you will come and go, geez, I've got this drop off of quality. I may need to go and do some trimming here. Now we have trimmed these reads, really just showing you, just to show you how, there's trim tails, there's trim front. There's lots of options for us here. Now we've trimmed these reads, we need to get them back out to something we can work with. We can use the write FastQ function. So we have read FastQ at the very beginning. We have write FastQ, and we just have a short read queue object here. We just tell it we want to write to FastQ.guz. I should write this out and zip this at the same time for you. Right? So we don't need to just write out a standard FastQ. It should gzip this for us at the same time. So in this entire session, we have been looking at a FastQ, which is subsampled, right? So 100,000 reads in a file. Okay, so often we will have 10, 100 times that many reads. We still need to be able to deal with them. And we have some solutions in Bioconductor which really revolve around either just taking a sample from your file, like we've done, and using that to be a representative of your file. We do QC on that, and we assume it represents your whole file. Or we can do things like FastQ Streamer, where we're going to take parts of your data at a time, process it, write it out. We'll take the next part, process it, write it out. So for evaluating quality of the FastQ, and this is what maybe if anyone's done any FastQ work before, there are tools like FastQC. This doesn't use all your reads, right? It takes a sample of your reads, and with that sample, it then gives you the quality. So the FastQ Sampler function allows us to specify how many reads we wish to sample when we're reading a file. So we use the FastQ Sampler function. Here, we give it our FastQ object here, sorry, file here. We tell it the number of reads we want. And that's going to create for us a FastQ Sampler object. Actually, at that point, we still have nothing. And when we run this function yield, then that's going to return an object of 100 reads from this file. Now, the really important difference between, say, FastQ Sampler and just taking the first 100,000 reads in a file is to do with the nature of FastQ. FastQ Sampler is going to read through your whole file and take 100 randomly, right, through that file. If you were to take the first 100 in your file, in your FastQ, typically FastQ, as it comes off the machine, is in the order of where it is in the flow cell, typically. This can cause problems if you only looked at the first 100 and you had a problem, you know, in the bottom right of your flow cell, but you're only looking at the top left. You're not going to see that. It's going to be totally lost on you. If you sampled across your whole FastQ, you will get a representation of all across the flow cell, and then you should be able to capture any of these artifacts, any of these potential problems. What you will do if you actually want to process the whole file is you will use this FastQ Streamer. And FastQ Streamer is just going to read in 100 reads at a time in the order. And every time I say yield, it will give me 100 reads. And then if I do another yield, it will give me the next 100 reads. And that's the importance, is the next 100. So it's keeping a reference to where you are in that file as you work along, right? In combination with a loop, you could do FastQ Streamer. I could take here 25,000 reads at a time. I say while, and here I'm going to yield in my loop, while the length of my object I yielded is greater than 0, while I still have reads left to actually process, print the length of the actual reads we've just yielded. Okay, and it will go through, and in this case, I guess it's divisible exactly by 25,000. So we get 25,000, 25,000, 25,000. We're done. Great. So I have one more thing I wanted to show you, and we haven't had time to really include this in this year, but I wanted to bring reference to it. So Wei in my team and JD, but Wei is the main author here. He really did a, you know, we were slightly frustrated by some of this streaming because it's really big files. It is going to take a little bit longer than you would like. So we sat down and we found a lovely piece of C software called FastP, and me and Wei learned a little bit of C, just enough in order to get this working from within R. What is kind of nice about it, it's not as interactive as what we would have, what we've just witnessed, which I think is very interactive. But it is very fast. So if you ever wanted to just run your, get some idea of the quality of your data, it's a new package, so we would definitely welcome feedback. Here it is, rfasp. You just need to actually, I'm just going to save this file. I'm going to stop and share my screen. So this is, I'm going to save that for a second. But if I do library, here I've got rfasp. It's just another Bioconductor-like package. Very simple tool to accept. Again here, the FastQ. It's not going to read the data into R for you. It's not going to return anything, an object. it's going to pass this file down to the C, and then return out a report. I'm going to give it. It's also going to return any trimmed FastQ, cleaned up FastQ for me. It's going to do a little bit of work. It's very quick. But what's really lovely is that we get a, hopefully, we're going to get a HTML report, which I'm going to stop share. If we open, it should give you a summary of your quality. So like that QA report I didn't display, you can really just give your FastQ in one line, and it's going to give you all the information we've just been going through. 76 basic cycles, it was single end. It's going to show you the quality drop off over time. It's going to show you the different makeup. There's some GC bias in your data, how your A, C, G, and T. This wasn't particularly biased data. Okay, so just a little example. If you wanted to, you can also check out our rfasp there. What version of R are you on, Jean? Okay, well, Jean's asked a question here. rfasp is not available on your version of R. So me and Wei, we only released this, I think, in the last release. So it should be in 3.12. So the version we're working through in our courses, it will be available. It's available on all systems. Usually what that is that your version of R perhaps is either a little bit older and you're not on version 3.12 of Bioconductor. But let me know, that would be interesting. Last thing, we do have some exercises for our session. If I can find our session. If you want to go through these, I don't think we're going to have time to go through them together, but if you have any questions, you can come on the issues and ask them. If anyone wants to run through them, just reach out on the chat and I will find some time in our sessions today. We can work through these. Okay, thank you for the session, guys. I will see you back here, I believe, at 12 o'clock.